I think it's pretty solid advice.
Groups of cells in different parts of your brain, they're all connected.
The statistics just don't back it up. There's nothing here.
Any complex life you find has mitochondria.
We have discovered things from athletes' juicing.
My name is Matt Kaeberlein, and welcome to the OptiSpan podcast. Hey everyone, welcome to the OptiSpan podcast. On today's episode, I'm going to talk about a recent paper that has gotten a lot of attention in the field and also picked up by the popular media. And this paper has to do with a treatment around inhibiting a molecule called IL-11. And so what I thought I'd do is start with a pretty quick and short introduction to the paper. I'll give you my take-homes, and then we'll go into a deep dive for those of you who really want to get into the data and really think about what it might mean and how it fits into the larger context of the field. So the paper was published in Nature in the middle of July, and it's titled Inhibition of IL-11 Signaling Extends Mammalian Health Span and Lifespan. This paper got picked up quite a bit by the popular media. Lots of stories were written. I kind of like this one, the supermodel granny drug could be secret to longer life, scientists say. This was actually a pretty reasonably written article, kind of a funny headline. Had this great picture of control aged mouse that looks sort of gray, kind of going bald. And then this age-matched IL-11 mouse where IL-11 levels were reduced that looks younger, looks skinnier, has nice, shiny, sleek black fur, can really show you visually the effect of IL-11. Now, I will say, I've seen a lot of these kinds of pictures, and it's very easy to cherry pick animals when you want to present this kind of data. So never really want to get too excited based on these kinds of pictures, because you'll see all sorts of stuff that's that's selectively modified to make you believe what they want you to believe. In this case, the data look pretty good, but in general, don't get too excited about these kinds of pictures that people throw up there. Okay, so my take-homes on the paper are, first and foremost, the data look pretty solid. Like, this is a high-quality paper. I think the authors did a pretty good job of interpreting their data well. The real take-homes are IL-11 is what we call a proinflammatory cytokine that appears to increase in expression with age, and that's true across multiple tissues. So we see this thing go up with age. It drives inflammation, and reducing IL-11 in a couple of different ways. They did this both genetically and with an antibody treatment, increases lifespan in mice. Again, the data look pretty solid, and appears to improve multiple health span metrics in mice. The biology here makes sense. It acts in the same pathway with our friend mTOR. mTOR, of course, is the mechanistic target of rapamycin. Rapamycin is a drug that inhibits mTOR. IL-11, in many ways, appears to be mimicking the effects of rapamycin. So the mechanism here actually makes a lot of sense. The effects on lifespan look good. They actually look almost identical to the effects of rapamycin. And I will actually show you the data in a few minutes to back that up. So again, the biology here all is making sense. This seems to be a new way, maybe a better way, maybe not, but a new way to tweak the same network that rapamycin tweaks when we treat cells or animals with rapamycin. So again, it all kind of lines up. I think it's real. I think it's solid. I think it will be reproducible. There are a few reasons not to get overly excited about this result at this point. So again, as I've sort of already alluded to, it's unclear if this is mechanistically at all different from rapamycin. It might be. I think there's reason to believe that it may tweak the network slightly differently, but I don't think there's any reason to think it's fundamentally different than rapamycin. And one implication of that is that we're likely to end up with similar effect sizes and maybe a similar risk-reward profile to rapamycin. Again, I'll talk about it a little bit when I do the deeper dive. There may be some reasons to think that the side effect profile could be different with IL-11 inhibition. Will it be better? I don't know. The authors try to kind of argue that. I think the claims about no side effects are overstated and probably incorrect. They didn't really look. So it's hard to really have a lot of confidence that there aren't any side effects when you don't actually really try to look for side effects. I suspect there will be. Will they be fewer than rapamycin? Maybe. I think we're just going to have to wait and see. Another reason why I'm not like jumping up and down about this paper is the magnitude of effects are really only about half what we know is possible with caloric restriction. So again, this isn't what I would call a game changer for the field. It is a new, therapeutically intriguing approach to tweak the same network that we already know affects longevity. So it may be more translational, but it's not fundamentally changing the way we think about the biology of aging. It's not going to get us to fundamentally new levels of health span or lifespan extension, in my view. It's also just the first study. So again, we always want to see replication. You have to ask the question, will the shiny new object stay shiny when more people are looking at it and working with it? Again, my view is this looks like it's real. I expect it to be reproducible. Will the magnitude of effect stay the same when more and more people start looking at it? I don't know. We'll have to wait and see. But so far, there's really only one lab studying this. And so we need to see more people working on it and getting similar results. So overall, promising. Effect size is good. Looks about as good as rapamycin, maybe a little better, maybe not quite as good. Maybe it'll unseat rapamycin as the gold standard eventually if it replicates, but it's not as good as caloric restriction in terms of magnitude of effect. It's still early. It's still a little bit unclear if this is fundamentally different or better than what we already knew about. Again, I don't think it is. I think it's targeting the same network. Downsides are still TBD. There are people out there walking around who apparently are knockouts for IL-11. I think there are opportunities to study those people and ask questions. Are they healthier? Are they living longer? Do they live 25% longer? I don't know. That is an answerable question. So it will be interesting to see. I don't know how common this is. I think there probably are going to be natural variations in circulating IL-11 levels in humans. Again, easy opportunities to ask, are those strongly correlated with health span and or lifespan or mortality risk? So I expect we'll see that data coming out. And again, if the signals look very strong and very positive, that will give us a lot of confidence that this is an important modality for modulating health and longevity in humans. There are also current clinical trials, both directions actually, increasing IL-11 levels or inhibiting IL-11 signaling. The inhibition, which is what is reported to extend lifespan in mice here, with an antibody treatment has been tested in phase 1 studies. Again, safety looks pretty good, nothing super concerning so far. And we will learn a lot, I think, from those ongoing clinical trials where IL-11 is being inhibited with monoclonal antibody therapies in humans. One thing I want to say is I think it would be premature for any of you to go online and try to buy an anti-IL-11 receptor alpha antibody, which is what was used in the paper, and inject yourself with it. Don't do that. Really, really don't do that. We don't know yet. This is one paper in mice. I really would not recommend that you try to adopt this therapy quite yet. I also would suggest that you not believe anybody who starts claiming or selling supplements that they claim reduce IL-11. I haven't actually looked. I suspect if that's not already happening, it will soon. There are all sorts of charlatans out there who will try to convince you that their supplement reduces IL-11. Don't believe it. At a minimum, they should be able to show you that they can measure IL-11 levels in blood from people taking their supplement and it goes down. In the absence of that data, just completely ignore them. Even if they've got that data, I think you'd really want to take a close look at it. Not clear yet that that would be therapeutically beneficial, but I doubt anybody actually has that data. So really, just there's so many snake oil salespeople out there. Don't fall into the trap of believing the opportunists who are just going to jump on this and claim that their supplement reduces IL-11. Just don't, would be my advice. Okay. Last thing I'll say, I don't think this needs to take a decade for us to figure out whether this is relevant in people and therapeutically useful in people. It probably will, just because that's the way things work, but it doesn't need to. I actually think this is almost a perfect example of an intervention where you could rapidly test this in companion animals, companion dogs in particular, assuming it's safe, which so far it looks to be in mice and the data we've got in people. The mechanisms work the same way. Antibody therapies work in companion animals, veterinary medicine. If you've ever heard of Librela, that's an example. So we can do this kind of therapeutic in companion animals pretty easily. We could know the answer 3 years in dogs if somebody was to step up, set that trial up, fund it, and do it. So we could get the answer there pretty quickly. Again, I think there's a reasonable chance it would work. We could also look in humans pretty quickly if people would start to do smart clinical trials in geroscience. And again, I think oral health makes a ton of sense as one place to consider looking. The endpoints are easy, non-invasive, and I think you can know the answer within a year. Easy. If you were to look at effects of geroscience interventions on oral health, periodontal disease. We already know rapamycin in mice can reverse periodontal disease, starting to get some evidence in people. And interestingly, there is evidence that IL-11 plays a role in periodontal disease. So this is like an obvious thing. If people would just figure this shit out and do the clinical trials, we could know these answers. So I don't think it needs to take a decade. I hope it doesn't. And I'd like to see some people actually get it together and do these clinical trials quickly. Because again, I think we can find out. We don't have to wait a long time to get to the truth. Okay, so that's the short take on this paper. Now, if you're interested, we'll do a little bit deeper dive. As always, the way I suggest doing this is start with the title and the abstract. Make sure you understand the abstract, every sentence in the abstract, and then ask yourself as you're going through the paper, do the authors actually show what they're claiming in the title and the abstract? And so we're going to start with the title. The title of the paper, again, is Inhibition of IL-11 Signaling Extends Mammalian Health Span and Lifespan. Okay, so only thing I'll say about this, and again, I think the authors did a really good job in the way they wrote the paper, and the abstract doesn't overstate or overhype the results too much. I do take a little bit of an issue with people claiming to extend health span. And look, I fully raised my hand back in 2016. If you look at the title of our rapamycin paper, we claim to extend health span. I'm reformed. The reason why I don't think people should claim to extend health span in the scientific literature is there is no consensus on how to measure health span. We do not have a way to quantitatively measure health span. Scientists should not claim to be able to increase or decrease or extend or shorten something that we don't have a way to measure, because you can't do statistics if you can't actually measure it. So it is scientifically invalid for people to claim that they have extended mammalian health span. Conceptually, I think we all get what they mean. They measured a few health span metrics. They saw things go in the right direction. They are able to statistically support those claims, but they did not show that they can extend mammalian health span. They did show that they can extend mammalian lifespan because we can measure lifespan. There is no ambiguity about lifespan. So again, it's a minor point. People do it all the time, but I would really like to see scientists be more precise and responsible in their language in this field. So, okay, that's get off my podium here and get back to the paper. Okay, what about the abstract? So the abstract starts with the sentence, for health span and lifespan, ERK, AMPK, and mTOR1 represent critical pathways, and inflammation is a centrally important hallmark. Okay, yes, if you're familiar with the hallmarks of aging, there are 12 of them. Inflammation is one of them. And then ERK, AMPK, and mTOR1. So ERK is extracellular signal regulated kinases. AMPK is AMP kinase. And mTOR1 is mechanistic target of rapamycin complex 1. Okay, yes, all have been implicated in aging. mTOR, clearly a central hub in the network that modulates the hallmarks of aging. We've talked a lot about rapamycin on this channel. Rapamycin is a drug that inhibits mTOR, gold standard for longevity or health span drug in animal studies. No question about it. AMP kinase, a ton of literature linking AMP kinase to longevity in different organisms. AMP kinase and mTOR talk to each other, so they signal to each other. They're in the same network. Again, lots and lots of data for AMP kinase being involved in aging. And interestingly, mTOR you turn down, and that seems to slow aging. AMP kinase you turn up, and that seems to slow aging. ERK, not quite so much evidence linking it to aging. It's in the same network, and we'll get into the network in a few minutes, believe me, but not at the same level of AMP kinase and mTOR. So we'll talk more about ERK and how it functions and how it may fit into the larger context, but nowhere nearly as well represented in the longevity literature as mTOR and AMP kinase. Okay, so we'll come back and do a little bit of a deeper dive on ERK in a few minutes. Second sentence here is, here we examined whether IL-11, a pro-inflammatory cytokine of the IL-6 family, has a negative effect on age-associated disease and lifespan. So they're just setting the stage. In this study, we're looking at IL-11. IL-11 is this pro-inflammatory cytokine, and we'll talk more about what a cytokine is in a minute. And they're asking whether or not IL-11 has a negative effect on aging biology. So what is a cytokine? Cytokine really is just a broad category of small secreted proteins. So they end up in circulation. They're circulating throughout our bodies, and they tend to regulate immune function, particularly inflammation. And the way they do this is they bind to receptors that are facing outward on the cell. So again, they're floating around in circulation. They come across the cell that has the right receptor. The cytokine molecule will bind that receptor, and then it'll signal through those receptors to change things inside the cells. And we'll get into a little bit more detail on this. Whole bunch of different types of cytokines. If you've ever heard of interferons or chemokines or interleukins, interleukin is what IL stands for in this case. Colony stimulating factors, tumor necrosis factors, all of those are different types of cytokines. Okay. Again, essentially they regulate immune function, also cell differentiation, so cell fate, cell proliferation. And they signal through this complicated and very interconnected network of intracellular proteins that are called kinases. So kinases here turn out to be very important. What kinases do is they add phosphate groups to other proteins. This is called phosphorylation. And that addition of a phosphate group is the signal. So as we'll see in a minute, you have these networks of kinases that signal in sort of a cascade to go from outside of the cell to actually doing some work inside the cell, often in the nucleus, regulating what are called transcription factors that then turn on or turn off genes, okay? But it's these serial phosphorylation events that are really doing the work. And there's lots and lots of different types of kinases in the cell. Our old friend mTOR is a kinase. That's what it does. It phosphorylates other target proteins to send a signal. mTOR is particularly responsive to nutrient levels. And so it's signaling when nutrients are high, you want to do one type of work. When nutrients are low, the cell wants to do a different type of work. And that signal is mediated through these phosphorylation events. Okay, we'll come back to kinases in a minute, but ERK is a kinase. And it's part of this complicated web of signaling that tells the cell when to do certain things and what to do in response to different factors in the environment. Okay, so how do these cytokines work? And I mentioned they bind to receptors. And there's this interesting sort of commonality in the way that these receptors come together. So often you will have this extracellular molecule, in this case the cytokine IL-11, floating around outside the cell. And IL-11 binds to something called the IL-11 receptor. And sometimes there's different flavors of these receptors. There might be an alpha and a beta. So in this case, we're interested in IL-11 receptor alpha. And IL-11 binds to IL-11 receptor alpha, which is in the membrane. It's a protein also. And it has a co-receptor called GP130. And the way this is a very common pattern in biology. You have two combinations of the IL-11 receptor alpha and the co-receptor GP130 come together with two IL-11 molecules on the outside of the cell. And it's that complex then that sends the signal. Okay, signal detected. Now we need to change something inside the cell. And it activates these kinase cascades. The canonical IL-11 kinase cascade is mediated through STATs. So STAT1, STAT3, it does a whole bunch of stuff: proliferation, survival, migration, invasion, differentiation, self-renewal. Again, kind of like mTOR, these kinases can do it all depending on the signals that they're receiving. But IL-11 can also signal through ERK, and it's ERK that the authors are focused on in this paper. And we can actually ask a question, and we'll get back to this. How much evidence is there that it's really ERK that's doing the work downstream of IL-11 in this paper? I would say it's mixed. It's not super convincing in my view that it's really ERK that's mediating most of these effects. It might be. Okay, regardless, IL-11 can signal through multiple kinases to do its job, and it's this structure of two receptors and co-receptors and two IL-11 molecules all coming together that are sufficient to induce the signal. So why did I take you through this? Again, this is a very common mechanism in biology. It's not even unique to cytokines. So this is common in other hormonal signaling cascades as well. And one that I think is interesting is a hormonal signaling cascade that I talked about with Crystal Hill recently on the podcast with the starvation hormone FGF21, or fibroblast growth factor 21. So as you will recall from that conversation with Crystal, in response to starvation or protein restriction, we get an increase in FGF21. FGF21 binds a different receptor, the FGF1 receptor, and its co-receptor in this case is beta-klotho. So you get the same sort of combination, 2, come together. That signals through a different kinase cascade, CAM-KK2. And part of the reason why I'm taking you through this is both to show you the commonality of structure function here, but also AMP kinase is downstream of FGF21, as is mTOR. I want you to start to appreciate how these networks are interconnected, because that's going to be important, I think, for understanding the biology here. So very, very different context. We're talking inflammatory signaling in one case, protein restriction or starvation in another case, common structure function signaling, overlapping downstream targets, and in both cases, we have a longevity readout, okay? So there are multiple ways to signal this network of interacting proteins mediated through kinase signaling to impact longevity. It's complicated, and I'm going to make it more complicated in a minute. All right, so that's IL-11, pro-inflammatory cytokine. Next sentence in the abstract. As mice age, IL-11 is upregulated across cell types and tissues to regulate an ERK, AMPK, mTORC1 axis to modulate cellular, tissue, and organismal level aging pathologies. Okay, so what they're claiming here is that IL-11 goes up with age. We'll look at that. That as it goes up with age, it changes signaling through ERK, AMPK, and mTORC1, and that those things are acting in the same pathway. And then the third claim here is it's that change in signaling that's actually mediating effects on aging biology, so lifespan and health span metrics. And I'll just tell you, the first two of those, to my mind, look pretty solid. The third part, I think, is plausible. It's probably right. I don't think there's a lot of evidence in this paper to support the idea that that's the mechanism specifically for how IL-11 is affecting lifespan and health span. Again, I think it's probably partly right, but I actually think it's going to be more of a network effect and really maybe mostly at the end of the day mediated through mTOR. But that's speculation on my part. There's not a lot of evidence in this paper one way or the other. Okay. The next sentence is deletion of IL-11 or IL-11 RA1. That's the receptor alpha, IL-11 receptor alpha 1. Protects against metabolic decline, multimorbidity, and frailty in old age. Administration of anti-IL-11 to 75-week-old mice for 25 weeks improves metabolism and muscle function and reduces aging biomarkers and frailty across sexes. Okay, so what they're saying here is that they have multiple different ways of reducing IL-11 signaling in the mice. And in all of the cases, you see what appear to be beneficial effects on health span metrics and or lifespan, and they actually look in several of the models at overlapping readouts. So this is great. Like, actually, this is one of the strongest parts of the paper to my mind. They have two different genetic models, and they have I'll just call it a drug model, this monoclonal antibody model where they can reduce IL-11 signaling. And in all three cases, they can show benefits on health span and or lifespan. So the two genetic models, one is the IL-11 gene itself knocked out. So you just don't make IL-11. The other is the receptor alpha gene knocked out. So you plenty of IL-11 circulating, you just don't signal through the receptor. And then they also have this monoclonal antibody. The great thing about the monoclonal antibodies, it kind of blocks the IL-11 signaling. It's like a sponge that soaks it up. The great thing about that, though, is you can actually start the treatment late in life. So you don't have any effects from having low IL-1 during development. You can start the treatment in middle age and see whether or not you get benefits on health span or lifespan, which they show that you do. That's important both for understanding whether it's a developmental effect and potentially for therapeutic translation. We're much more interested in something that you can start treating people or companion animals with in middle age, as opposed to something that you have to treat people starting in their teenage years or maybe even earlier than that to get the effects. One thing I will say, they don't have an ERK depletion model here, so it doesn't, again, make the strongest case that this is actually mediated through ERK. Okay, in lifespan studies, genetic deletion of IL-11 extended the lives of mice of both sexes by 24.9% on average. Treatment with the anti-IL-11 antibody from 75 weeks of age until death extends the median lifespan of male mice by 22.5% and female mice by 25%. So we'll look at the lifespan data in a minute. Basically, they're just claiming they could extend lifespan both with a genetic model and then, as I just mentioned, with a midlife treatment with the antibody, which again is potentially much more therapeutically, translationally useful if we want to start thinking about moving this into humans or companion animals. End of the abstract is, together, these results demonstrate a role for the pro-inflammatory factor IL-11 in mammalian health span and lifespan. We suggest that anti-IL-11 therapy, which is currently in early stage clinical trials for fibrotic lung disease, may provide a translational opportunity to determine the effects of IL-11 inhibition on aging pathologies in older people. So again, making the case, I think rightly so, that this is potentially translationally useful. Again, I really like the way the authors wrote the abstract. I think it's stated clearly. I think they don't overstate the results in general. So kudos to the authors for doing a good job there. I do want to say the very first figure now is this model where they are trying to make the case that this is IL-11 through IL-11 receptor alpha and GP130 through ERK, then signaling to this AMPK mTOR cascade through a kinase, LKB1. Again, we'll come back and talk about kinase signaling again in a minute. So this to me is really overly simplistic. Again, as I'm going to maybe hammer harder than you want me to, this is a complicated, interconnected network. This model, I get why they did it. I'm not saying the model's wrong. It is just, in my view, way, way too simplistic for what's probably actually happening here. Nonetheless, this is the model they wanted to start with to try to set the stage for what they think is going on. For those of you who aren't used to looking at these kinds of models, just the arrows are sort of what you would expect. That means you're activating. So if we look at IL-11 activating through the receptor in GP130, that activates ERK and STAT3. The other symbols here, which are in red, look like a sort of a T-shaped thing. Those mean the opposite. They mean you're inhibiting or turning down. So when you turn ERK up, you turn down LKB1. LKB1 is an activator of AMP kinase. AMP kinase is an inhibitor of mTOR. Okay, so it's just directionality. That's what those symbols mean for those of you who might be wondering. Okay, so what do we know about ERK? As I've sort of already alluded to, ERK is a kinase, extracellular signal regulated kinases, also known as MAP kinases, mitogen activated kinases. And MAP kinases have been studied extensively for decades, right? These are sort of the canonical signaling molecules in the cell that are involved in sensing all sorts of stuff outside the cell and telling the cell what to do in response to those signals through these phosphorylation events. They're involved in a huge number of things. ERK is part of the RAS signaling pathway. Some of you may have heard of RAS. It's a very famous cancer gene. If you get hyperactivation of RAS, that signals proliferation, which can signal different types of cancer. RAS signals through RAF to MEK to ERK. Those are all kinases now in this signaling cascade. Okay, so ERK is what the paper is focused on. The reason I'm showing you this, starting with this first very simple diagram, is I want to make the point this pathway has been implicated in longevity for a long time. To the best of my knowledge, I think RAS was first implicated in longevity by Michael Jaswinski all the way back in 1990, where he showed using a yeast model of aging that RAS could affect longevity in yeast. More recently, Linda Partridge's lab has shown that a drug that inhibits the kinases just upstream of ERK, MEK1/2, the drug is called trametinib, can also extend lifespan. First, they did that in flies. Kathy Slack did that when she was a postdoc in Linda's lab. And then more recently, they showed in mice that treatment with trametinib or trametinib plus rapamycin could increase lifespan in mice. So pretty good evidence already that this pathway has been implicated in longevity in at least 3 different model organisms that are used to study aging in the laboratory. In that paper where they looked at trametinib in flies, I really like this particular diagram because, again, it makes the point about crosstalk between these kinase signaling pathways. So in flies, this is all downstream of the insulin-like receptor where you have, again, this RAS, RAF, MEK, ERK signaling, trametinib inhibiting MEK to increase lifespan in flies. One of the reasons why I like this diagram is we can just point to all of the things in this diagram that have been previously implicated in longevity, right? So because this is all part of and connected to the insulin signaling pathway, we've got the kinases in that pathway, AKT, FOXO. Again, this is the classic insulin IGF-1 pathway from C. elegans, famous from the work of Cynthia Kenyon, all the way back, again, in the 1990s, as a highly, highly potent and conserved regulating pathway for longevity across many, many different species, even in people. So FOXO, the transcription factor that sits at the bottom of this pathway, that's DAF16 in worms, has variants in humans that are strongly associated with longevity. The insulin receptor in humans also associated with longevity. So lots of reasons to believe that this, now we're starting to talk about a network, right? So pathway would be one side of this, but there's interactions here. Lots of reasons to believe that multiple components of this network can implicate longevity. And I'm going to keep making this a little bit more complicated. Again, I think it's useful to understand how interactive these things are. And part of the reason why I do that is so you recognize overly simplistic models when you see them. Okay, so this is another way that people have sort of tried to diagram out the MAP kinase signaling pathways. I mentioned these phosphorylation events. So you've got the receptors. So you have an extracellular molecule, IL-11 in this case, but there's a bunch of different molecules that can do that, binds to their receptors, signals to these kinases. So at the top here are what people call MAP kinases. So RAF, which is in that RAS signaling pathway I just showed you. There are others. They become activated, become phosphorylated. They then phosphorylate the next kinases down the pathway that are called MAP kinases, okay? So that's where MEK1/2 comes in the ERK part of this network, right? MEK1/2 then becomes activated. It phosphorylates ERK1/2, and then those kinases can go phosphorylate other kinases or go directly do activities on transcription factors, things like that. So we talked about LKB1. That's going to be a next kinase downstream of ERK1/2. So these things are sort of presented as, parallel linear pathways, but there's all sorts of crosstalk between them. It's also worth mentioning all of the MAP kinases, the bottom level there, have been implicated in longevity in different animal models. So multiple parts of these MAP kinase signaling pathways that have been implicated in lifespan regulation in mostly invertebrates, but some data in mice as well. So well characterized components that are important for longevity. Here's another way to look at the network that's a little bit more complicated. Again, just to kind of let you start to feel how these things interact. Highlighting here those MAP kinases, ERK, JNK, and P38 in this little bit more complicated network diagram. And here's an even more complicated network diagram. And even this is an oversimplification. But I think we're going to stop here. We don't want to make it too much more complicated. And part of the reason why I like this kind of a network diagram is you can then start to ask yourself, how are these things interconnected and how many of these things have been implicated in regulating lifespan or aging? And it turns out a lot. So here's ERK in the network and our other friends, mTOR complex 1 and AMP kinase. Again, going back to this simplistic diagram that was in figure 1 of the paper. Again, you can see the connections here aren't quite as simplistic as they're made to be in that paper. And there are lots of stuff talking to each other. And if we look more broadly in the network, there's a bunch of stuff here that has been implicated in regulating longevity in different organisms. And mostly here, I'm talking about things where you can increase lifespan. And this isn't a comprehensive list. Like, I didn't go look at every single component of this network and ask, can I find some reason to link it to aging? This is just what off the top of my head, because I'm passingly familiar with the broader literature in the field. I can say, yep, I know that one's been shown to increase lifespan. Oh yeah, that one's been shown to increase lifespan. So this is not even, I'm sure I could find more red circles to put up there. This is just to give you a feel for how strongly connected this network is to the biology of aging. And the way I think about it is, it's really this network that underlies the hallmarks of aging. That's what we talk about when we're talking about speeding up or slowing down biological aging. We're talking about tweaking this network. The way I think about it is, there are just certain nodes in the network that are more amenable to turning up or turning down to have significant effects on lifespan and health span metrics. Again, maybe I'm completely wrong. I don't think I'm completely wrong. Maybe that's too simplistic, but I think it fits pretty well with the data that's out there. And it turns out that some things like mTOR are just really useful nodes in the network for tweaking the rate of aging. Not everything in the network is that useful. There are some things that if you turn them too far up or turn them too far down, you end up dead, right? Or they don't actually change the network very much when you turn them up or turn them down. But other things are quite sensitive, and you can actually have a pretty significant impact. So that's kind of the way I think about the biology of aging right now. I think there's lots of stuff outside the network we don't understand yet. We need to study that. But what we're starting to see is most of the things that most people in the field are studying, you can pretty easily fit into this network. And I think that's where IL-11 falls. It might be a really useful node in this network for modulating the biology of aging. Okay. So let's go back now and actually take a look at some of the data. I'm not going to go through every figure, but I do want to take you through some of the data so you can get a feel for, what's actually shown and how strong the data is. So again, we'll start with the first part of the abstract. As mice age, IL-11 is upregulated across cell types and tissues to regulate this ERK, AMPK, mTORC1 axis, or I would say this network, to modulate cellular tissue and organismal level aging pathology. So let's look at the first part of that. They're claiming that IL-11 is upregulated across cells and tissues in the body with age. Okay, so the way that they do this is they're measuring the abundance of IL-11. That's shown in the top line here. The darker the bands, the more IL-11 that was detected. The second line down, GAP-DH, is what's called a loading control. This is what people will typically refer to as a housekeeping gene. The assumption is that the expression level of the housekeeping gene won't change with whatever condition you're looking at, age in this case. That is an assumption, so you do have to always ask yourself, is it really the case that the housekeeping gene doesn't change? But if we assume that that's correct, then you can actually make sure that you didn't accidentally load a lot more or a lot less total protein into the well of the gel, which would be an artifact in this experiment. So what you really are interested in would be the ratio, say, of the band for IL-11 to the band for the housekeeping gene. In this case, most people will just look at it by eyeball. You say, okay, the housekeeping gene didn't really change with age. That means that a change that we see in IL-11 probably real. Okay. And what you can see pretty convincingly is that IL-11 amount goes up with age. In this case, they're looking in liver between 12 weeks, 25 weeks, 50 weeks, 75 weeks, 110 weeks. Okay. So it goes up with age. And let's see, 110 weeks is probably about, 75-year-old biologically mouse. So they've got a pretty good age range here. Okay. Now, for the other things where you see that p, little p, lowercase p before ERK12 or P90 RSK, they're not looking at total protein levels. They're looking at the phosphorylation of these kinases. Again, this is supposed to be a measure of the activity of those kinases as opposed to their abundance. What you would really want to also see is the total amount of the kinases, because oftentimes what you're really interested in is the ratio of the phosphorylated form to the total amount. They don't show total amount here. So we'll just accept for the purposes of now that this is a rough activity level assessment for these kinases. And again, what you can see pretty clearly with age, ERK activity goes up, LKB1 activity goes up, mTOR activity goes up, and then P70 S6 kinase and PS6RP, that's S6 ribosomal protein, are downstream of mTOR. So those are targets. S6 kinase is a target of mTOR. S6 ribosomal protein is a target of S6 kinases. So these are things that if mTOR activity goes up, you'd expect to see those go up as well. Okay. Interestingly, the S6 kinase knockout mouse is a little bit long-lived. It's not as long-lived as you can get with rapamycin, but in and of itself, it is a little bit long-lived. So reason to believe that S6 kinase, at least, also may play a direct role in longevity. So the case here looks pretty convincing. Those things all increase in activity with age. IL-11 goes up with age. AMP kinase phosphorylation goes down with age. Remember, I mentioned before, more AMP kinase activity is associated with better longevity, better health span. Less mTOR signaling, turning down mTOR activity, is associated with better longevity, better health span. So arrows are all going in the direction we would expect, given what we kind of know from the literature about this network and signaling through these molecules. Okay, so yes, looks like IL-11 is upregulated with age. Looks like the ERK, AMPK, mTORC1 activities are going in the expected direction. They don't actually show here that this is causal, but we'll get to that in a minute. Okay, what about across tissues? So they look in 4 different tissues, see the same pattern as we go from young to old, an increase in IL-11 levels. Fortunately, in the IL-11 knockout, that's the IL-11 minus mice, we don't see any IL-11. I would be worried if we saw IL-11 in the mice that are supposed to be lacking IL-11. So that's good. So, it's not showing that IL-11 goes up in every tissue, but in a small subset of tissues, it looks like we see this general trend of an age-related increase in IL-11. So what about the causality question? And here, this is a nice experiment. So what they've done is they've now looked in both the wild type and the receptor alpha knockout mice. So this is a different genetic model where the receptor is deleted. And they're only, in this case, comparing 10-week to 110 weeks. So they didn't do the full age time course. But if we just look at young versus old, what you can see is that for the most part, not completely, but for the most part, the age-related changes in signaling through ERK and AMPK and mTOR go down, or they're attenuated in the receptor alpha knockout mice. Again, it's not complete. So this is consistent with the idea that part of the effects on mTOR and AMP kinase and LKB1 are mediated through IL-11, part of the age-related effects, but not all of the age-related effects. So that's the argument for a causality. Another thing that's interesting here is they look at markers of senescence. So P16 and P21, there's some other data on senescence in the paper as well, showing that senescence markers go up with age. And again, that's mostly dependent on IL-11. So if you get rid of IL-11, you see a pretty dramatic decrease in both P16 and P21. So this is making the case now that part of what IL-11 is doing is it's driving age-related senescence, or at least expression of these senescence markers. So I think this is pretty convincing that IL-11 goes up with age, and that increase in IL-11 mediates some of the age-related changes that we see in AMP kinase and mTOR and other downstream targets. I will say, particularly this first blot, when I looked at it, my gut reaction is, wow, that's a really clean blot. Like, you don't really see blots that look this clean. So I suspect, this experiment was done lots and lots of times, and they took the best looking blot. But this data looks really nice, which is good. But I also start to get a little bit worried when the data looks a little too nice. I don't think there's anything wrong here. But that's just kind of, I think, something that people who've done a lot of biology and read a lot of papers sometimes pick up on. You're like, ooh, that looks like a really clean blot. So this looks pretty clean, but it does look convincing. And again, I think this is a solid result. Okay, so what about the causality for longevity and health span, though? Because that's the last claim in this statement here, and I don't think there's really anything in the paper that supports that causality. It's more plausible, very reasonable, given especially that we know that mTOR is a potent modulator of longevity and health span in mice, but no direct evidence that the longevity and health span benefits that we're going to get to in a minute are directly mediated by mTOR, AMP kinase, LKB1. So that's a little bit more speculative. It's okay. I'm not saying the authors should have had those experiments in there. That would be like a future study, but it is worth recognizing that's a little bit of an overstatement that they didn't actually show in the paper. Okay, so what about evidence for age-related health span changes? I'm not going to take you through all the data. Again, I'm just going to tell you it looks pretty good. I believe that reducing IL-11 levels, and they did this multiple different ways, 2 genetic models, the monoclonal antibody treatments, reduces body weight. So body weight gain with age was prevented, reduces fat mass, seems to preserve lean mass. That's pretty interesting. So body composition going in the right direction. Some evidence for frailty. Again, I'm not a huge fan of these mouse frailty studies. I don't think there's a lot of consistency across the literature yet, but in general, looks like function is preserved in a few different ways with reduced IL-11 levels with aging. I thought the improved insulin sensitivity was interesting. So this is a case where it might be a little bit different than rapamycin. Rapamycin treatment in mice is commonly associated with, at least in the context of an oral glucose tolerance test, perturbed glucose homeostasis and reduced insulin sensitivity. So this may be a case where IL-11 is superior to rapamycin. I don't think there's a ton of evidence that that's a negative with rapamycin, but that's often the way people interpret it. So this might be a case where IL-11 is a little bit better, at least for metabolic homeostasis, than rapamycin treatment. Grip strength seemed to be better. That was normalized to body weight, so you have to be a little bit careful in how you interpret that because body weight's less. One interesting thing was I thought the higher body temperature in a couple of the models, they showed that reducing IL-11 levels led to higher body temperature. And there's an explanation here in that I think one of the gene expression studies they did, they saw that one of the most highly upregulated genes was something called UCP1 or uncoupling protein 1. Uncoupling proteins allow protons to flow across the mitochondrial membrane, and that gives off heat. So this might explain the increase in body temperature and also the positive changes in body composition and the fact that they reported the IL-11 depleted mice actually ate more but had a lower body weight. And there's kind of a famous drug called dinitrophenol that was actually used as a weight loss drug. It's an uncoupler. It was pulled from the market because people ended up dying from taking it because too much uncoupling is bad. But it fits. It makes a pretty, again, plausible biological mechanism here that could explain the changes in body composition, the changes in body weight, change in body temperature, maybe also associated with some of the lifespan effects. So I think that's an interesting thing for future studies to look at. How much is that metabolic effect contributing to health span and potentially lifespan outcomes in these animals? And do we see it in people? Again, there are humans walking around with IL-11 knockouts. Do they have higher body temperature? I don't know. That's an easily measurable thing. Like, we should be able to get the answer to that question if it's not known. Okay, hopefully we'll get that answer pretty soon. So again, I think there's a lot of really interesting data here. And I think there's going to be some very cool follow-up studies that do a little bit of a deeper dive into some of these questions. Okay, so what about lifespan? So again, if we go back to the abstract, in lifespan studies, genetic deletion of IL-11 extended the lives of mice of both sexes by 24.9% on average. Treatment with anti-IL-11, that's the antibody, from 75 weeks of age until death extends the median lifespan of male mice by 22.5% and female mice by 25%. Okay, first thing is, we always got to be careful about percent changes. Percent changes are influenced by the absolute lifespan of the control animals. If the controls are short-lived, you get an artificially inflated percent change because the denominator is smaller, and that leads to a larger number when we look at percent change. So have to be careful with percent changes. So I can tell just by looking at these curves, it looks pretty good. So the effects look real. The controls look reasonably long-lived. Again, this is the pooled data for males and females. The data is in the paper for each sex independently. It worked in both sexes. So we're going to just take a little bit of a deeper dive into the pooled data. Again, for those of you who don't look at lifespan curves all the time, I just want to give you this really easy rule of thumb for evaluating, does this look real or not? That rule is called the 900-day rule. It is very simple. So this is a preprint that we put out with Brian Kennedy and Camille Pabis and colleagues, where we looked at a bunch of data that from published mouse longevity studies and came up with this 900-day rule. And the rule is, again, very simple. In the absence of independent replication, a putative mouse longevity intervention should only be considered with high confidence when control lifespans are close to 900 days, or if the final lifespan of the treated group is considerably greater than 900 days. That's it. We want the controls to be close to 900 days. What does close mean? I don't know. 850 to 950, right? Ballpark. Or, preferably, and we want the treated group, the supposedly long-lived group, to be much greater than 900 days. What's much greater? 1,000, 1,050, right? That's a real solid result. Why is this important? This is important because the literature is absolutely full of claimed lifespan extension in mice from interventions where the controls were short-lived. 700 days, 600 days, 500 days. Believe it or not, sometimes 400 days. The controls were that short, and then they claim that intervention X increased lifespan. And then when somebody came back and spent 3 years, 4 years, 5 years trying to reproduce that experiment in reasonably long-lived control animals, there's no effect. One explanation is the putative effect was really an artifact of the fact that your controls were way too short-lived. That's why this is important, because mouse experiments take a long time to do. It's going to be a while before we have replication. You want to have some way to assess confidence in, is this a real result or is it an artifact of something that went wrong in the experiment? There's all sorts of stuff that can go wrong. Okay, so that's why we use the 900-day rule. Now let's apply it. How do we apply it? We're going to start with the y-axis. This is true for every survival curve, assuming it's plotted this way. We're going to start with the y-axis. You're going to go halfway down. That's either going to be 0.5 or 50, depending on whether they're doing a ratio or percent survival. Go halfway down, draw an arrow straight across till you hit the curve that you're interested in. In this case, it's the controls, that's shown in black. As soon as you hit the curve, then go straight down to the x-axis. Wherever you hit the x-axis is the median lifespan of that group. So the median lifespan of the control animals in this experiment was 850 days. That's pretty good. Remember I said close to 900. That's close. So that looks solid. And the effect on top of that also looks pretty solid. It's a big effect. We can do the same thing with the treated group. Start at 0.5, go over to the curve, go down to the x-axis. We get about 1,050 days. That looks pretty darn good. So I'm impressed now. I'm like, okay, this looks real. That's a large effect. That's comparable to what we can see with rapamycin. And I'll show you the data in a minute. What about with the monoclonal antibody experiment? Remember here, they started the treatment in middle age. They started the treatment at 75 weeks of age. Again, that's, I don't know, 60-year-old mouse, biological. Controls look good, about 850 days again. Treated look good, a little bit longer even, 1,085 days. Again, that's an estimate. I'm just ballparking, eyeballing, right? Obviously, that may be off by 10, 20 days. Okay. Again, this looks really solid to me. This is better than 95, 98% of the mouse lifespan experiments that you're going to see in the literature. I'd encourage you to go back. If you have a favorite intervention that you're super excited about, go back to the literature and see how it stacks up. Okay. So we can actually do that. And I like to compare it to a couple of experiments. The first experiment is a study from my lab that we published in 2016, where we treated mice with rapamycin for 3 months. And then we just asked, what is the effect on lifespan? They were treated from 20 to 23 months. So it's not perfectly apples to apples, but it's pretty close to the monoclonal antibody experiment here, where they started treatment at 75 weeks of age. That's almost 20 months of age. Okay, so we can just overlay the data with the prior experiment from 2016. The black here are the controls. The red are the rapamycin-treated animals. You can see nice long-lived controls in this experiment. And if we put the IL-11 monoclonal antibody data right on top of it, you can see it looks comparable to 3 months of treatment with rapamycin. So is it better than rapamycin? I don't think there's any data here to suggest that, but it's comparable. That's actually pretty impressive. There's not much else, maybe nothing from a drug perspective in the literature that's comparable to rapamycin. So this is not bad. So I think this looks like a real result. It seems robust. The controls were appropriately long-lived. The treatment group is as good as we can get with rapamycin, or at least as we have gotten with rapamycin to date. So that looks very promising. The other thing I do is I like to compare results to a sort of classic caloric restriction experiment that Rick Weindruck and Roy Walford did back in the 1980s, where they looked at 65% caloric restriction and its effects on lifespan in mice. And what you can see is neither rapamycin nor the IL-11 monoclonal antibody are comparable to the magnitude of median or maximum lifespan extension that was achieved with 65% caloric restriction. Why do I do this? It's not to depress people, but I think it is important to recognize that this is not what we would consider really a game changer in the field. We're not going way beyond. We're not breaking any records here. And I really think it's important for people to appreciate that, because you have people out there sort of making nonsense claims about longevity escape velocity, right? If we can't do better than a lifespan experiment that was done almost 40 years ago, I don't think there's much evidence to support the idea that we're getting close to longevity escape velocity. I think we need to be realistic. I also think this should be a wake-up call to people in the field that we probably need to start looking outside of the hallmarks of aging to figure out what else is out there if we really want to move the needle. Okay, so that's all I'm going to say, but it is useful to appreciate where this stacks up compared to a couple of the better effects that have been seen in the literature in terms of longevity in mice. Comparable to rapamycin, nowhere near the magnitude of effect that's possible with caloric restriction. Okay, that's kind of my deep dive on the IL-11 paper. Again, I think it's a very solid paper. I think the take-homes are IL-11 increases with age in mice, and reducing IL-11 increases lifespan and improves multiple health span metrics in mice. IL-11 pretty clearly acts on the known longevity network, and that includes mTOR signaling. The effects on lifespan are comparable to 3 months of treatment with rapamycin in old age. May have a better health span profile. I think time will tell. I think certainly there's reason to believe that IL-11 is tweaking the network in a slightly different way than rapamycin, and that could lead to both better effects in some contexts, worse effects in other contexts. I think we just need more research to figure that out. The effects aren't as good as CR. I think the side effect profile is really still TBD. This is only one study from one lab. We really need to see, I think the results are going to hold up. I think we need to see whether or not the magnitude of effect holds up. Maybe it'll get better. Maybe it won't be quite as strong going forward. So time will tell there. I also think there's some pretty good reasons to be optimistic about the clinical applicability or translational applicability of this therapy. So it's still early in the clinical pipeline, but there are already therapeutics around IL-11 antibody treatments that are being tested. Actually, there's studies going on both directions. Increasing IL-11 levels in people for certain conditions, targeting IL-11 with an antibody similar to what was done here in humans. The early phase 1 data from the IL-11 depletion with antibody looks good. So I think time will tell, but there's reason to be optimistic that this approach may be viable in humans. There are also people walking around with these IL-11 mutations that have either dramatically reduced or zero IL-11, so we know it's not lethal, at least. And I think we have opportunities to learn from those people. Are they healthier? Are they potentially living longer? Those are answerable questions. I'm pretty optimistic. This at least gives us another shot on goal, maybe a better therapeutic option ultimately than rapamycin. So that's it. Hopefully you found this deep dive on the IL-11 paper informative. As always, if you've got any questions or comments, please feel free to leave them below. If you're not yet a subscriber, hit that subscribe button. And I hope to see you next time on the OptiSpan podcast.